So I'm going to tell you about our work on dilated myopathy and restrictive myopathy and therapeutic target discovery. And this work is motivated by the problem that, as you know, heart failure is a tremendously serious problem, and there are relatively few pharmaceuticals for treating heart failure, and they typically target not the etiologies of the disease, but rather the symptoms of the disease and make it easier for the heart to work, in essence. So what we've set about to do is look for a screen for therapeutic targets and in essence, launch early-stage drug discovery pipeline from that. We focus on inherited models of cardiomyopathy, and typically dilated myopathy, but more recently restrictive cardiomyopathy, as I'll tell you about. And the reason we study inherited disease is because we use iPS cardiomyocytes as the model system. And so we are able then to reproduce the phenotype of, or at least some in vitro manifestations of the disease in the dish that we can then study. So I'm going to tell you first about modeling dilated myopathy using iPS models. I'll move on to talking about similar studies with restrictive myopathy. And then lastly, what I'd like to do is talk about experiments that came out of studies about almost a decade ago now, looking for molecules that might be useful for regenerating the heart after injury. Okay, so dilated myopathy, as you know, about a third to half the cases can have a genetic component to the disease. And it's those the monogenic forms of the disease that we're interested in because we can create iPS cells and, as I said before, reveal aspects of the disease in the dish. The problem is that current therapies largely address the symptoms of the disease and make it easier for the cardiovascular system to function, but don't address the underlying reasons for the disease. Now, in inherited disease, you might expect that there would be multiple mechanisms. So what we're thinking about is that this disease might become not just one disease, but there might be multiple diseases and therefore multiple therapeutic options depending on the underlying genetic lesion for inherited DCM. So what we do is large-scale screens based on iPS models, and just to reiterate or recapitulate what iPS cells are, these are cells that can be derived from any tissue in the body. We just do a blood draw. We convert those cells into iPS cells, which for all intents and purposes resemble embryonic stem cells or cells of the early implantation embryo. And they can be directed through judicious use of cytokines to different tissue types that we can then use in our studies. And so how we measure contractility is illustrated on this slide. You can see, actually, how do I point on the slide with the pointer? Can you see that? No. Okay, I'm just going to mention it then. Okay, so how we measure contractility is that we put the cells, we mature the cells, they form nice two-dimensional arrays of tissue. They're in 384 Weill plates. We can then monitor the motion of the cells. In more sophisticated assays that I'll tell you about a little bit later, we actually we put them on a soft substratum and we can look at the deformation of the substratum underneath. And because we know the material properties of the substratum, calculate the forces they generate. And so we can derive a lot of metrics about the contractility of the cells. So to illustrate our approach, I'm going to tell you start with a story about dilated myopathy due to mutations in the phospholamban protein. The phospholamban protein regulates the calcium reuptake pump that takes calcium back into the sarcoplasmic reticulum with each cardiac cycle. So patients with the mutation in phospholamban in arginine 14 deletion mutation present typically in the second to third decade of life with an arrhythmogenic cardiomyopathy. So we derived iPS cells from a patient, and then we corrected those iPS cells using CRISPR. We've also introduced the mutation into a healthy donor's iPS cell, so we ended up having multiple isogenic pairs. And the isogenic cells serve as a control for the patient cells. So as you can see in this slide, the contractility is diminished in the patient line, and it's diminished also in the healthy donor line when the mutation is introduced relative to its own isogenic control. So in this experiment, in this project, we took a fairly straightforward approach. We sequenced the hell out of it, and we looked for things that might be altered. And we asked, are any of these processes that are suggested to be altered by sequence possible therapeutic targets? And what we came up with from all of this sequencing is that the unfolded protein response is upregulated in this form of dilated myopathy. So to delve a little deeper into this, there are 3 branches to the unfolded protein response. So it recognizes proteins in the endoplasmic reticulum, and if they're misfolded or unfolded, it tries to refold them. If that doesn't work, it will actually kill the cell. So you could imagine that this process would be good, or it could be a good process to upregulate, maybe compensatory, or it could be bad if too much of it's going on and it's killing the cells. So what we found by knocking out each of these arms is that basically you made the disease worse if you knocked out these arms. In particular, the third arm, this PERK branch here, if you knocked it out, you got the most profound effect. So we figured that the signaling through that branch was therapeutic for this disease. So following that up, we asked whether you could stimulate the UPR with a small molecule. This is not a drug-like compound. This is just a reagent molecule. Could you stimulate that pathway, and would it be beneficial for the disease? And as you can see here, if you treat with that small molecule known as BIX, it will restore the function in the cells that carry the R14 deletion to the level of the wild type. And it has almost no effect on the wild type cells or the healthy cells. And so from that, we think that we're onto something here, that if we could pharmacologically target this pathway, this would be a mechanism-specific therapy for phospholamban disease when they carry this R14 deletion. So what we've learned from this is that ER stress activates this PERK pathway, activates the transcription factor ATF4, and that upregulates the unfolded protein response. That's therapeutic. So the ATF4 itself is a really interesting transcription factor. It had been shown previously that ATF4 is beneficial in the context of pressure overload heart failure in animals. So we thought we were really onto something with ATF4, and what we wanted to understand was a little more about what ATF4 is doing that's relevant for this disease. So at the time, we were conducting a large-scale chemical biology screen. We were probing all sorts of kinase inhibitors and phosphatase inhibitors, trying to map pathways that would be beneficial for this disease. And what we came up with were a couple of molecules that seemed to be beneficial, and we used them as chemical genomics probes to study mechanisms. And to make a long story short, what Isaac Perea and Dries Feyen did in the lab was they were mapping the signals downstream of these 2 probes that would be important. These probes activated ATF4, and those probes activate downstream enzymes involved in de novo serine metabolism. So what is de novo serine metabolism? So from glycolysis, basically from phosphoglycerate, what you have is a shunt going from phosphoglycerate towards serine and 1-carbon metabolism. And what we realized hooking up with Christian Metallo, who's at the Salk, who does all this beautiful carbon mapping, is that pathway is upregulated in this form of dilated myopathy. Downregulated in this form of dilated myopathy, and efforts, if you activate it through ATF4, that's therapeutic. And that was actually foreseen in the literature before. Activation of serine 1-carbon metabolism has been shown to be beneficial in myocardial hypertrophy in animal models. And then there's a recent paper that just came out is in bioarchives that it acts through affecting mitochondrial disease, which we thought was really interesting, and I'll get back to in a few slides. So our overall view of this process now is that ER stress activates this PERK pathway, which is a good thing, activates ATF4, and works through these downstream processes, perhaps to compensate for mitochondrial dysfunction in the disease. Okay. This is great. This was a good story, but it took a long time, a number of years, a number of postdocs' lives. And the question is, we still have just this tip of the iceberg. We hardly understand the disease. And so I really wanted to do work in high throughput. And so our technology has all along been trying to move these assays towards high throughput so that we could get a bigger picture of what might be therapeutic target space. So a ex-postdoc in the lab, Francesca Briganti, who just took assistant professorship position at University of California, San Diego, led this effort in the lab. And she used iPS lines from different patients. We took a few different genotypes, you'll see on the next slide. And then what she did was large-scale screens, functional genomic screens, to knock out genes and asking then what genes, if you knock them down, will restore function, that as we read it out in vitro. And then the idea is to take those forward into animal models. So these were large-scale proteomic screens scanning most of the proteome. So here was the actual experiment that she did. She took a couple of different genotypes, troponin T, mutant DCM, RBM20, mutant DCM, and the phospholamban mutant DCM. And she used degenerate probes that were based on microRNAs, so that each probe sees multiple targets, and then we deconvolute the target space. And I can get into that in detail if anyone is interested how we do that. So she ended up with lots of probes that went into the funnel. 2 probes look really promising, and that led to 203 targets, of which individual inhibition of 117 would restore contractile function. And 30 were as good as CRISPR correction of the underlying genetic lesion. And so those numbers are down here, and actually about 5 of them are pathways that have been studied in these types in heart failure models in animals. So what do these proteins do? So our informatics colleagues map them to a tremendous amount of processes in the cell, but ER stress comes out prominently, so we're intrigued by that, but we see other processes as well. And mind you, I forgot to say this, but those probes do nothing to isogenic control cells, so this is all disease specific. So from this, we now have a slightly expanded view of that module I was talking about. We think AMPK fits into this through NERF, and then NERF regulates ATF4, the molecule I was talking about previously, and ATF4 then activates the different processes that I was talking about. So we're now exploring chemical space all around the activation of NERF and ATF4 in order to alleviate, and figuring out the best way to alleviate the contractile deficit in these forms of DCM. Okay, so AMPK is very interesting to us, and so as if you look in the literature, small molecule activators of AMPK have actually had a beneficial effect. There was a paper not too long ago out of Germany on this. So we think we're on the right track, and what we need to understand now is how to drug this pathway. Okay. So this is what I've told you about, is that ER stress through this pathway we think is beneficial. And we've published a couple of the papers that a couple of the studies that I've talked about, and the large-scale screening is unpublished work. Okay, so I'd like to shift now and talk about restrictive myopathy and where we are with that. So we're interested in restrictive myopathy because it's, in essence, an inherited form of disease that exhibits diastolic dysfunction. And as you know, diastolic dysfunction can account for about, underlies HFpEF, which accounts for half or a little over half of heart failure cases that we see today. And so we're very interested in having a genetic model of diastolic dysfunction to look for what might be therapeutic. So to introduce pediatric early onset restrictive myopathy, these images were acquired by a pediatric cardiologist in the lab of one of his patients, and so you can see the blown out atria and so they have elevated filling pressures of the heart. So how to model this disease? So we're using a variation of the approach that I talked about before, but what we do is we image calcium and contractility, and we actually now are measuring contractility by looking at deformation of the substratum under the cells. And so when we take images in sequence, so in interleaved images, where we get calcium and contractile data. So from that, we generate calcium contractile curves, and you can actually put them together in a relationship. It looks a little bit like a PV loop, but what it really is basically the calcium contraction cycle with each heartbeat. And that can give you calcium sensitivity because you can see how much squeeze or how much squeeze or relaxation you get at different levels of calcium. Okay. So to measure diastolic function, you, however, need to know the floor of your assay. So if we put the cells onto a substratum, the substratum has the beads that we're tracking, and those cells will give a baseline level of diastolic tension, and then with contraction, that will contract further. So we can tell the baseline level of tension by comparing the gel with no beads with the gel during diastole with the cells on. I'm sorry, the gel with no cells versus the diastolic frame. And that can give you this basal level, this diastolic tension. Okay, so what this looks like in practice is shown here. So here we get a healthy donor cell, and you're getting nice curves, and you see the floor and you see the ceiling of each contraction. So you have force on the y-axis and time on the x. So if you have restrictive myopathy due to a mutation here, what we see is that not the ceiling is raised, but so is the floor, right? And you can see that the net amount of tension generated between the floor and the ceiling is less. So we think this is an in vitro model then of diastolic dysfunction. The floor is high, the contraction is high, but importantly, the net squeeze that you get is reduced. And on the opposite, this is dilated myopathy due to another mutation in troponin I3. And here you see basically the systolic function being tanked, basically. The floor is down, but of course the ceiling is way down too. Okay, so armed with this assay that we can now have diastolic dysfunction in a dish, we're able then, and this is quantification of it, so you can see that there is an increased maximal force, a decreased diastolic in the HCM, RCM variants, and there is decreased forces in the DCM variants. Okay, so armed with this, we can try to disambiguate variants in ClinVar. And so we've made a chassis, a cell that doesn't have any troponin I3, troponin I1, and now we take lentiviruses and we put back in troponin I3 with different mutations. We want to disambiguate, go through the entire ClinVar, but what we did is we took about 45 variants as just to work out the assay. These are the initial library that we did. This was a medical student, Peter Tran, in the lab, and a visiting undergraduate, Dongzhu Han, did this experiment. And so what they find is if we look at calcium sensitivity as the metric, because that's actually the underlying problem with thin filament disease that we're looking at, what you can see is that to the left, these are the controls here, and to the left are all the variants that have DCM or have RCM or HCM, so restrictive or hypertrophic, to the left, and to the right, over here we have the definitive dilated myopathy with the reduced contraction. Okay, so does this correlate at all with human clinical data? And indeed, if you look at this calcium sensitivity metric and you compare it to age of onset of disease, you can get a pretty good correlation with an R-squared value of like 0.4. If we go back into our database in the Center for Inherited Cardiovascular Diseases at Stanford, and we look at patients that have those mutations, there are many fewer, of course, but what we find is a really nice correlation of about, we looked at the age of cardiac cath, and we now get a 0.8 correlation using our data. So can we use this for drug testing? Could we embark on a screen? So we took a couple of different molecules. We took a myosin inhibitor, mavacamten, and we took a calcium desensitizer, W7, to ask which of these would be therapeutic. Could we distinguish them? And you can see a dose curve for MAVA on the left, so the myosin poison, basically you can see the systolic function on the left of those curves being basically decreases dramatically, as well as the diastolic tension decreases modestly to the level you'd like. But mavacamten seems dangerous because, of course, it would tank the systolic function. The calcium desensitizer is a little better, might have a better therapeutic index. And so by looking at this in more detail, if we look at the best doses and we just look at those here, you can see that you can get some therapeutic benefit in terms of decreased diastolic function without horribly tanking the systolic function. So we think for thin filament disease, where the calcium sensitivity is the issue, approaching the calcium sensitivity directly might be the best therapeutic approach. We've not looked at phosphodiesterase inhibitors, which are of course used in clinic, and there's a lot of interest there because different ones can have beneficial or no effect. So that's something we want to explore. Okay, so what I've told you about then is that we've explored RCM variants, we've generated the model for disease, we have a platform now to disambiguate the ClinVar variants and to do drug screening. So am I over time? I have what? I am over time. You're okay, go on. Oh. You were a little ahead. Oh, okay. So if I'm allowed to continue then, I'd like to give a few slides, 2 minutes left, okay, on what is a really exciting project for me, and this goes back to screens years ago looking for regulators of cardiomyocyte replication in the hope that they might be therapeutic for diseases where acute myocardial infarction, maybe chronic ischemic disease. And so this was a project that my wife led in her laboratory at Stanford, and before she left to take this into a company. So years ago, she had the hypothesis that there will be some protein made by the epicardium, and it would stimulate the regeneration of the heart. She basically fractionated media, found that that protein is follistatin-like 1. It promoted replication, and when put back into mice, it would restore cardiac function after acute MI, and that's what you see in this slide, and that was published back in 2015. What is follistatin-like 1? So, and the cells that are regenerated are from pre-existing cardiomyocytes. So what is follistatin-like 1? So follistatin-like 1 is a small adipokine cytokine. It's made by the fat and skeletal muscle with endurance exercise, and it has beneficial effects on the cardiovascular system. But in us, we make it after infarction, but it's not regenerative. But it's heavily, heavily glycosylated. The fish form is not so glycosylated, and it is indeed important for regeneration in the fish. And the form that's regenerative in humans lacks glycosylation because it was produced in bacteria. So we set up a company called Regencor to focus on the unglycosylated form generated by mutating the sites for glycosylation. And when we put that into mice, rats, or pig models of acute MI, it indeed improves function after MI, and it is regenerative. It increases muscularization, reduces scar. And these are studies shown with pigs that are genetically predisposed to diabetes and metabolic syndrome, so it's not just healthy young pigs. And you can see here that there is a reduction in the necrotic area, there's a reduction in the scar volume, and there's improvement by invasive hemodynamics in systolic function and diastolic function of the heart. Okay, so then, this is my last slide. We think that these effects are likely to translate to humans because we see several key things. We see a reduction in scar size, we see a decrease in volume, and we see an increase in ejection fraction. And these three things in meta-analysis of registrational trials have shown the predictivity of clinical outcome. So we were encouraged by this. And now we're poised to try to raise some money for this company, which is not an easy task in this environment. So I'll stop there, acknowledge my supporters in this work or the funders of these projects, and thank you very much for your attention.